There we go. All right. Burdette is online. Newton, Massey. Oh, yeah, Massey's here. 2.5. Richardson, Cedric, Rice, here in red. Pauly, Midkiff is here in red. Eric, Sipple, Hodges, David, Kip, Hudson, Felder, Tudor, here in red. Bowers, Hemmings, Asbury is here in red. Redman, Reed, Michaela is here in red. Kinsey's here. Van Cross. Bowers is online. Hodges is online. Cedric is online. Eric is online. And just to remind you guys, you're getting all extra credit just for being here for the rest of the semester. If you're physically present instead of online, you get extra credit. I'm trying to encourage people to physically come in, because the online thing was supposed to be like, oh, I'm not feeling good today, or oh, I overslept. The occasional. You can do it all for the whole thing online if you want, but it helps to be here. For some people, maybe not for others. You better than I do. Some students, you might just zone out when you're here, right? And maybe when you're online, you're forced to pay attention. I don't know. Do your thing. Just be honest with yourself. Less than a minute. Does anybody have any questions? My first announcement, I guess, to let you guys know, my office hours will be sort of cut short today. I've got a department meeting at 10, so I'll be in my office until 10. And then after that, I'll still be online, so you can still reach me online for office hours, but I'll be in a meeting. So what else? Take a look at the next week's lab if you haven't already. The entire thing is virtual, so we will meet next week, or you bring your computer in. But if you get it done early, at the very least, you might get some extra credit, and you might not have to come in. So we'll see. It'll help you to just do it early, I think. But again, it's up to you. All right. Well, it's 8. I guess we'll go ahead and get started. Wait, good time. Richardson just showed up online. Might as well wait till 8:01. Does anybody have any plans this weekend? Anything worth mentioning? Nope, me either. Well, that's nice of you.
No, I know what afterwards.
Where does she live?
Crosslanes.
Oh, that's good. So it's not like you have a long drive ahead of you.
Oh, no.
Good. All right, now it's 8:01, so we will officially get started. Jump back into chapter 9. So to remind you, this is the second chapter that will be on the next exam. So it'll be chapter 8, 9, and 10 for exam 3. Where are we at? There we go. Yes. So the first word for attendance, let's see, is there anything up here worth using? Yeah, dominant would be the first word for attendance. So as usual, if you're in person, you can send those words for a little extra credit. If you're online, send those words before 9 a.m. If you're watching the video when I post it later, instead of sending me the word dominant, send me a picture of something that is dominant. And I don't necessarily mean, biologically. Like maybe I would have sent a picture of the 2012 Florida State football team because they dominated. Something like that, right? Be creative. Anyway, this is where we left off. But last, let me back up one step and remind you some of the terms I told you about. Homologous chromosomes, you already know from chapter 8, but we mentioned it again in this chapter. And the new term here was gene locus. And that's basically, it's what it sounds like. It's the location of a gene on a chromosome. And what I didn't say yesterday, because I was kind of rushed in this last slide, was that when I was a student, this used to confuse me because this thing pointed here, here, and here, and it says loci, which is, plural for locus. And then this thing also said allele. So I'm like, what in the world? What's the difference here? When I first saw this, I'm like, what is it? Is it an allele or is it a loci? But those are just two different things, right? Basically, all three of those are alleles, right? This is a dominant allele for gene P, both. A recessive allele for gene A for both. And a dominant allele for gene B and a recessive allele for gene B. So yes, those are all alleles, but then this is also happens to be their location, right? So like you could talk about houses. You've got a house at this address, a condo at this address, and a trailer at this address. So you're talking about different things, but you would still describe them as their address. So that's all that means. But more importantly, for understanding the information in the semester, you need to understand what an allele is. And I know we've talked about it a lot. You should be very familiar with it after the lab. But again, an allele is an alternate version of a gene. So whatever gene we might have for chin dimple, wherever it is on the human chromosome, it's right here for me, then it's going to be right here for you. And that's the whole idea of a homologous chromosome, right? The genes, all the genes are in the same places on the same chromosomes. There just might be different versions of those genes, but they're at the same place. So any questions about this slide? Oh yeah, and again, make sure you understand the difference between homozygous dominant, right, which would be this, homozygous recessive, and heterozygous. Very important, at least for testing purposes. All right, let's move forward. And we're still in the first main bullet point. It's a very long chapter. It's not that complicated, but it is very long. So what we just got finished talking about was Mendel's law of segregation, which basically says, when you make sperm or egg, when you go through meiosis, one allele for each gene goes into the sperm or egg, right? Or if we're talking about chromosomes, one homologous chromosome, right? So you've got one of each, one from each of your parents in your sperm or your egg. That's the law of segregation. Now we'll go back and talk about Mendel's law of independent assortment. And we talked about that in chapter 8, and we're talking about it again. So it's the same information. We're just going to look at it from a different viewpoint, different context. Before we can do that, we need to talk about a dihybrid cross. We already talked about what a monohybrid cross is, right? So that's basically what you did in lab. When you guys did Punnett squares to see what your hypothetical children would look like, that would be a monohybrid cross for each one that you did, right? Because you looked at one character at a time. Excuse me, I'll tie my shoe. A dihybrid cross, as you might imagine, instead of looking at one character at a time, you're looking at two characters at a time. And before I even really get into this, I'll just go ahead and do a little spoiler alert. Basically, what I'm about to get at is that genes are not linked together, right? They don't travel together because they're on completely different spots, right? Different chromosomes sometimes, or if they're on the same chromosome, they're on different spots of the chromosome. So anyway, again, a dihybrid cross is the mating of parental varieties differing in two characters. If, and this isn't the case, but if the genes for two characters were inherited together, then this is what would happen, right? The F1 would produce only the same two kinds of gametes they received from the parents, and the F2 would show that 3-to-1 phenotypic ratio. An easier way of saying that is if the genes for two characters were inherited together, then it would still be the same as a monohybrid cross. You would still have that 3-to-1 phenotypic ratio. And this will make hopefully more sense when I show you the picture. But that's not the case, right? Well, sometimes it is. Sometimes genes are on the same chromosome, but very often they're not, right? So they don't travel together. But if the two characters are sorted independently, which is what actually happens, then the F1 produces 4 different genotypes. And we just happen to use, in this example, we're using R's and Y's, and you'll see why later, equally. So then that means the F2 generation, right, that third generation, has 9 different genotypes producing 4 different phenotypes in a ratio of 9-to-3 to 3-to-1. So all this long-winded way of saying, which basically you need to know is for a monohybrid cross, like we've already discussed. In that F2 generation, you should see a 3-to-1 phenotypic ratio. And you should associate the 3-to-1 ratio with a monohybrid cross. For a dihybrid cross, like we said right here, you should see a 9-to-3 to 3-to-1 ratio. It's basically that simple. Your book makes it a little bit more complicated because it could be more complicated if we kind of dove into it and if we really paused and took our time here and made sure you really, really understood the intricacies of this. But in my opinion, it's not that important because you can have a weak understanding of this, but still understand the stuff I'm going to teach you later in this chapter and later in the semester. So for that reason, I'm going to move forward if no one has any pictures or questions about this slide before I show you a visualization of this slide. All right. So this is what I was basically saying. This is that what if situation on the left, but doesn't actually happen. And then on the right is what actually happens, right? So if the genes for P color, and not like urine color, but the color of the plant P color and P shape, if they were inherited together, then this is what you would end up with. You would still end up with that 3-to-1 ratio, right? Because if every time you had a yellow, it was smooth, and every time you had a green, it was wrinkled, that's what would happen. But they don't. That is not what happens because these alleles are separate. They're on separate chromosomes, right? So when they separate, when this thing goes through meiosis, those things are going to separate, right? So you have a big R, right? Or big R, little r, and then the Y's also separate. It even gets even more complicated, not in your book, but think about this. This is only a dihybrid cross. Try to picture like a trihybrid cross, or 4 or 5 or whatever. Again, this is a really complicated way of saying the genes don't separate together because of independent assortment. Therefore, you end up with all these different possibilities. That's all we're saying. So any questions about this slide? Again, it's just a visualization. It's that 9-to-3 to 3-to-1 ratio that you should recognize. And I will say this, you might have to do a Punnett square. I mean, this is a Punnett square. You might have to do a Punnett square to figure out some questions on the exam, but it would be a simple Punnett square like this, where only where only looking at one character at a time instead of one like this, where you're looking at 2 characters at a time. So again, when you're studying, don't feel like you need to memorize that big Punnett square. You don't need to memorize any of the Punnett squares, but you don't need to memorize any Punnett squares. You should be able to do a small one. I'm not going to ask you to do a big one. Unless I want to throw one in there for extra credit, but probably not that either. All right. So when you do a dihybrid cross, the result supports the hypothesis that each pair of alleles segregates independent of each other during gamete formation. So to remind you what we said independent assortment was in the context of chapter 8, remember we said all the chromosomes line up, the homologous chromosomes line up in metaphase of meiosis 1. And it's, let's put it like this. Of course, you have 23 pairs, and I'm only about to draw 3 of them. But remember, they all line up, those little X chromosomes, right? There we go. Doesn't look too bad, I guess. So we have those 3 pairs of homologous chromosomes. And it's not like they all line up. We're like, all right, those are all the chromosomes you got from your mom, and those are all the ones you got from your dad, right? That's not what happens, right? Independent assortment states what actually happens is it could be any which way, right? It could be mom and dad, and then dad and mom, and then mom and dad again. Or maybe it is all mom and then all dad, right? All the different combinations, right? And remember, with 23 chromosomes, as we know from chapter 8, that means there's 64 trillion possibilities when it comes to gamete formations. Anyway, so that's how we described the Mendel's law, or excuse me, the law of independent assortment in chapter 8. Now in chapter 9, we say this. Independent assortment is seen at, well, I'll get that in a second, sorry. Basically, yeah, here we go. The inheritance of one character has no effect on the inheritance of another because they're just completely different. Now, I'll give you a different example that's not true, but imagine if, again, the gene for, I don't know, stinky feet and smelly breath, that was on the same chromosome, right? And you have and your mom has sticky feet and smelly breath and your dad doesn't, right? So then when you go through and you produce your gametes, your egg and your sperm, then there is a possibility, right, because they're on the same chromosome, that you're either given your mom's chromosome, or you're definitely either given your mom's chromosome and your dad's, right? That's the way it works. They separate in meiosis. But then, because they are on the same chromosome, if you're given the mom chromosome, then you're going to have the allele for stinky feet and smelly breath, right? Meanwhile, the dad chromosome is going to have the alleles for not stinky feet and not smelly breath, right? Because in that case, they are on the same chromosome. So yes, they would be inherited together. But in most situations, they are not inherited together, and that is the law of independent assortment in the context of this chapter. And again, in the context of chapter 8, we're just saying they don't line up in any kind of specific way. It's all random. And like I said, the independent work topic, I wouldn't expect anybody to do it. You wouldn't have enough time in your college career to do it. But if you were to draw all 23 of your chromosomes, 23 pairs, and then try to draw one of each combination of all the different ways they can line up, mom and dad. And again, that's in the millions. So, you wouldn't really have time. And I certainly wouldn't have time to check it. Anyway, any questions about this slide before I give you an example using Labrador retrievers? Which used to be the #1 dog breed in the country, by the way. And recently it's a French Bulldog. So also, hey, there's an independent work topic if you want. Look into dogs. If you like dags, as they say in the movie, just write about dogs. Yeah, so French Bulldogs are the #1 right now. And they have a lot of health problems, which a lot of purebred dogs do, because basically purebred is another word for inbred. Not on purpose, but that's a whole different topic. We'll get to it if you want for independent work. Anyway, let's get back to these Labradors. So here we go. In these Labradors, the color black is dominant to the color brown, and being able to see is dominant to being blind. So we have these. Here we have a black dog that can see, so that's 2 dominant phenotypes. And also I'm going to put this in here because we'll talk a little bit about it later. We know this thing has 2 dominant phenotypes, right? Black and can see. We don't know its genotype. We know at least it has 1 dominant allele for the hair color and 1 dominant allele for vision, right? But we don't know what the other one is, right? Because with a dominant phenotype, as long as you have 1 dominant allele, you're going to show that phenotype. Meanwhile, we have this black dog that cannot see, right? So we know for that's the phenotype, right? Black, which is dominant hair, recessive vision, right? So we know, again, for the hair color, we know the genotype is a big B something, and that's all we know. We don't know what the other one is. Could be big B, could be little b. Doesn't matter. Either way, it's getting the dominant phenotype. Meanwhile, if you can't see, and that's the recessive phenotype, we know it has to be homozygous recessive. We know it has to be little n, little n. Because if it had a big N, just 1 big N, then it would be able to see. And of course, we've got a brown dog that can see. So again, in this case, it's homozygous recessive for the hair color, because that's a recessive phenotype. And then we know at least 1 big N for the vision. And then this one is homozygous recessive for both. So there we go. There's what we're going to talk about. I'm not going to take any questions yet, but we're going to talk about these dogs right here. Okay, so if you were to mate them, if you were to mate 2 heterozygous dogs, which is what you can see right here, right? So this dog, you can't see it, but we can see its genotypes, right? So looking at these genotypes, we know it's a black dog with normal vision, crossed with another black dog and normal vision. If they were to have puppies, statistically, that's what you should end up with. And again, there's that 9 to 3 to 3 to 1 ratio, right? So you would have 9 that are homozygous dominant for both, right? You would have 3 that are homozygous or dominant for one, and recessive for the other, so on and so forth. I'm not going to waste your time talking about all this. The point here is, again, you get this 9 to 3 to 3 to 1 ratio, which you should recognize as a dihybrid cross. And again, more importantly, the idea here is that those things are separate, right? The genes for coat and the genes for vision must be on different chromosomes. Because if they were on the same chromosome, we wouldn't get this 9 to 3 to 3 to 1 ratio, right? It would be inherited together. And you would only have that 3 to 1 ratio that we've looked at previously. So any questions about labs? No, not really. I'm not going to talk about labs, but any questions about independent assortment? All right. It's really a lot of talking for something pretty simple. right? The hardest part in the exam, I think, when it comes to independent assortment is the fact that independent assortment by name sounds a lot like segregation. There's a sorting and segregating, right? It sounds very similar. So just remember, for law of segregation, you're saying that there's only one of each allele that goes into the gamete, right? Because you have two of each alleles. And when you make your sperm or egg, only one goes into each one. And again, law of independent assortment is saying they line up independent of each other. They're not linked. Anyway, going back to the lab idea, let's talk about using a test cross to determine an unknown genotype. So there might be like one question on this on the exam because it's good to know. It's a good thought exercise. But even if you forget it, you'll be able to understand everything else for the rest of the semester. So for that purpose, this is an unimportant discussion, but let's have it really quickly. All right. The next word for attendance, I'm going to underline it. And be careful here because this word has two parts, and I only want this one part. That's the next word for attendance. So again, if you're online, make sure you open your eyes and look what I just underlined. That's the next word for attendance. If you're watching the video, not the live stream, but after I post the video later today or later this weekend, if you're watching the video, again, instead of sending me that word, send me a picture of one of those things. Anyway, let's talk about a test cross. A test cross is a mating between an individual of a dominant phenotype, but an unknown genotype, and an individual that's homozygous recessive, right? So basically, we have this dominant phenotype individual, and we don't know its genotype, and we want to figure out its genotype. Imagine if we did that with humans. Like, I really don't know about John's genotype. Hey, come here, Susie. Why don't you guys have sex real quick? I'm going to look at your kids and figure this out, right? Extremely unethical for humans, but for plants and a lot of animals, we do it all the time. So let's give you this as an example, right? Let's look at this, go back to these Labradors and talk about this. Like I said, this cute little guy right here with black hair and normal vision. Oh, okay, excuse me, we're only focused on the hair. We know that's the dominant phenotype, right? So because it's the dominant phenotype, we know it has at least one of the dominant alleles. We don't know the other because that could be a big B right here, or it could be a little b. Wouldn't change the phenotype. Unlike this dog, right? That's the one, no questions asked there. We know if that dog's brown, then it must have two recessive alleles. So to figure out the genotype of that black dog, then we do what's called a test cross. Again, we mate them. We look at their offspring. So here's the two possibilities, right? So first of all, if this was big B, because we don't know the other one, that would be this, right? So if this was big B, and you crossed it with a brown dog, then you would have all the offspring would be heterozygous. Therefore, they would all be brown, right? Meanwhile, if this was big B, then half of the offspring would be big B, or big B, little b, and the other half would be little b. Or more importantly for the phenotypes, because again, you can't see genotypes, you can see phenotypes. About half of the offspring would be brown, and about half of them would be black. That might not make much sense to you because for some reason your book just decided to get rid of an actual Punnett square and only give you a portion of it. So let's look at it this way very quickly. So again, if we're looking at this scenario, big B crossed with a little b. Oh man, I might not have enough room to write that. But hopefully you can see no matter what, all four of these squares are going to be big B, little b. Can you guys see that without me having to draw that out? No matter what, those are the only combinations. Big B, little b, big B, little b, big B, little b, big B, little b. So again, 100% of that, the dogs would be black. Meanwhile, again, I'll show you this one in an actual Punnett square. It's good to know this anyway, right? Because you got to do this for the exam. So again, this is the situation here. So we'll say big B, little b. So hard to do this. Little b, and then little b. There we go. So here we go. Big B, little b come together. Big B, little b come together again. So hard to do that. And then here we go, little b right here. Little BB. And then same right here, little b. So again, in this case, half of the offspring would be black and half would be brown. Of course, this isn't perfect. I don't know if your book talks about this. Not that this matters, because again, the only reason they've been taking this time right now is because test crosses are not important. But by talking about this, we're really enforcing this idea of homozygous dominant versus homozygous recessive versus heterozygous, and this idea of a Punnett square, and all that stuff is important. So anyway, this is just what is expected. It is technically possible for this thing to happen, right? This is what we would expect. We would expect 50% of them to be black and 50% to be brown, but it's all random. It's all by choice. It's all whichever egg meets with whatever sperm. So it is statistically possible, but improbable, for this mating to have 100% black dogs. In which case, you might look at that and say, "Oh, it's 100% black. So this dog must be homozygous dominant." But that's not the case, right? So basically, what I'm saying here is that you cannot definitely say, you can't definitely figure out that this thing is homozygous dominant. But if there are any brown dogs, for sure, if you have some brown dogs as the puppies, then you know for sure that it's heterozygous. So you can prove heterozygous. You can't necessarily prove homozygous dominant. Not that that's important. Labradors aren't important for this semester. Test crosses aren't important, but I want you to be able to understand this idea of dominant versus recessive and homozygous dominant, heterozygous, and homozygous recessive. So any questions about these puppies? Let's move forward. This is where I think this is the fun stuff to me, the rules of probability. And I think if I remember correctly, when you guys did your All About Me quiz for independent work, and if you haven't, you still can, a lot of you said that your hardest class was math. The bad news, here's a little bit of math. The good news is there's not going to be much of it on the exam. And the other good news is, hopefully in my mind, it'll be easy math. I'm not going to try to make it hard. I'm going to try to make it easy. Genetic crosses, so all these monohybrid crosses that I've been showing you, the Punnett squares, they obey the rules of probability. These are the same rules that apply when you're tossing a coin or rolling dice or drawing cards. I'm sure you guys know those rules, right? When you toss a coin, there's a 50% chance it'll land on heads, 50% chance it'll land on tails. When you roll a dice or die, if you roll just one, right, there's a 1 in 6 chance it's going to land on any number. If you pull a card, there's a 1 in 52 chance you're going to pull I don't know, any card you choose, right? Ace of Spades. Also a good song. Rule of multiplication is the probability of a compound event, or states that the probability of a compound event is the product of the separate probabilities of independent events. What? That sounds like some really confusing gibberish, but it's actually not. So when I talk about the rules of probability, I'm not going to ask you to define the rules of probability. I'm going to ask you to apply the rules of probability in a problem. So on the next slide, when I show you how it works, then hopefully you'll be like, oh yeah, that's a lot easier than the definition of the rules of probability. Actually, before I show you the next slide, I will give you an example. Like I said, the chances of getting any one card are 1 in 52. You guys understand that, right? Because there's 52 cards. So the chance of getting, if I say an ace of spades, is 1 in 52. Make sense? So 1 in 52 chance of getting an ace of spades. Now, if I were to say, all right, well, what are the chances of getting an ace of spades and a 2 of hearts? Right? Well, okay, there's a 1 in 52 chance of the ace of spades. There's also a 1 in 52 chance of getting the 2 of hearts. So the chances of those things both happening, and that's you multiply it. And I'm not a math major, so I don't know 52 times 52 off the top of my head. And the answer is 2,704. So the chances of that are 1,200 or 2,000, 1 in 2,704. And actually, I simplified this. Technically, this isn't correct because after you pull out the 1 card, and there's only 51 cards left, so technically it'd be 52 times 51, but we don't need to get into that. I just wanted to show you the basic math. So any questions on this slide before I give you a more biological example? All right. So here, this is a lot simpler, right? So when this female up here, this is a heterozygous female, when she produces eggs, right, those alleles are going to segregate, right? And when that happens, if you were to look at, like, let's say you're just like choosing, you've got all of her eggs in front of you. That's not possible, but well, I guess technically it is. But imagine you could just see all of her eggs, and you just pluck them out of a bucket or whatever. All right, so the chances of you picking up one with a big B is 1 in 2. The chances of you picking up with a little b is 1 in 2. Same for him. When he makes sperm, they're going to segregate. So the chances of you picking any one of his sperm and having a big B is 1 in 2. The chances of you picking any of one of his sperms and them being little b is 1 in 2. So, right, that's what these statistics are. The chances of a big B from her, 1 in 2. The chances of little b, 1 in 2. The chances of big B, 1 in 2. The chances of big little b, 1 in 2, right? So those are the chances of you getting this egg over this one, or this sperm over this one. But then what are the chances of this egg meeting with this sperm? Well, it's a 1 in 2 chance of having that egg and a 1 in 2 chance of that that sperm. So you multiply them. 1/2 times 1/2 is 1/4. And that's what you're going to need to do on the exam. There's going to be a question where I say, what are the chances of all right, this person mates and this person mates. What are the chances of them having a female with this disease? And I'm going to explain the disease part later. But you know the chances of having a male over a female is 1 in 2, right? It's a 50-50 chance. So if I say, let's say there's a disease where there's a 1 in 10 chance of getting it, right? And then I say, what are the chances of this couple having a kid with that disease that's a female? So then you would multiply the probabilities. 1 in 10 times 1 in 2 gives you 1 in 20. It'll be something like that. And again, I'm going to explain the disease part later, because I am going to teach you some ways to figure out the probabilities of having a kid with a certain disease, which is very relevant to your everyday life. Maybe. I know some of you, from some of the questions you've asked recently or answered, you don't want kids, so you won't have to worry about this. But anyway, any questions about this slide? All right. What your book doesn't really talk about, since they won't talk about it, I won't talk about it either. Let's not muddy the waters here. Let's just do another word for attendance. Since we're looking at these things, coin is the next word for attendance. So if you're in person, just send me the pictures. Actually, sorry to throw a wrench in your plans, but if you're online, whether you're watching the video now or the live stream now or the video later, I take back what I said earlier. So for the all the words, show me the pictures. So go back and take a picture of these things. So again, for a coin, don't show me a picture of this coin and don't send me the word coin. Send me a picture of a coin. All right. Anyway, any questions? Let's talk about family pedigrees. Now that we know about the idea of test cross, and we're very familiar with homozygous dominant and homozygous recessive and heterozygous, and the difference between the relationship between phenotype and genotype, and the rules of probability, we can bring all this together and talk about family pedigrees. And again, this is slightly relevant because you might have to worry about something like this in your family to figure out what are the chances of getting this disease or passing along this disease. Anyway, as your book points out, Mendel's principles apply to the inheritance of many human traits. So far, Mendel's all into P. Not like R. Kelly, but pea plants. Mendel's into pea plants. All the work he did was with pea plants. So what your book is saying here is that a lot of the stuff that I just taught you, the rules that I just taught you, also apply to humans. Some human traits, not a lot of them. So the next slide illustrates the alternative forms of three traits, I assume, thought to be determined by simple dominant recessive inheritance of one gene. So a lot of the stuff we talked about in lab basically is what we're talking about here, right? Well, we didn't talk about freckles. I think I might add that to the next semester. Anyway, freckles, according to this anyway, is based on one gene, right? You're either dominant or you're recessive. There you go. Widow's peak. You're either dominant or you're recessive. You either have it or you don't. Free earlobes. You either have the dominant or the recessive, right? It's either attached or it's not. Of course, again, this is just an example. So when you're studying, if you're looking at your book, don't be like, all right, I got to memorize. See, is the widow's peak dominant or recessive? I'm never going to ask you that, right? That's all irrelevant. We just use these things as examples to show you how genetics work. It's It's not important to know if widow's peak is recessive or not, for example, right? So these are just examples, but let's talk about it. As you already know, for example, from the lab, and I forgot which section it was, but one of you asked a great question. Was slightly confused, as I understood you would be. Like, why isn't the dominant one more dominant, so to speak, right? Why isn't the dominant like cleft chins? I think it must have been the Monday section. So there must have been like one person with a cleft chin, if I remember correctly, and everybody else had a smooth chin. But cleft chin is dominant. So it's like, well, why is that? Well, that's just the way it is. There's no real reason to explain it. But there is a word for it that you need to know. The wild type is the one that you see the most in nature. So, for example, right now in this room, how many of you have a cleft chin? Okay, again, so she has the dominant phenotype, but the dominant is not the wild type in this case. There's a lot more people. I don't have a cleft chin either. There's a lot more people who don't have it in this classroom. There's an independent work topic if you want to, now that I'm thinking about it, because now I'm curious what that is in humans. Like, is it such an overwhelming smooth chin to dimple chin? You could look that up for any of the traits that we talked about in lab. What is it? Like, are there more people with freckles or more people without freckles? Are there more people with widow's peak or more people without? Or does anybody know? So you can look that up. You do your own little experiment if you want. If you can't find the information, go find it yourself. Like, according to my samples, just go look at some people. Go stalk some people on Facebook. It is a book of faces after all. You just go look at some pictures and just tally your data, right? That would be an experiment. You have your hypothesis, you're collecting data, and then you're going to analyze your data to see if your hypothesis is correct or not. Anyway, any questions about what a wild type trait is? There'll be one question on that on the exam, so make sure you understand dominant doesn't necessarily mean more common, because the word for the more common one is called wild type. All right, let's move forward. So how can human genetics be studied? We know for peas, because what Mendel did and what we talked about what Mendel did, he'd take a flower, cut the male parts off, right? And he'd say, all right, this flower is going to mate with this flower, and he would make it happen. We can't do that with humans, right? It's not ethical to be like, all right, Susie, I want to study eye color, so I want you to have sex with John, and I'm going to take your kids, and I want those kids to have sex with each other, and then I want to look at their kids, right? You can't do that. It's unethical. So how can we do it? Well, geneticists analyze the results of matings that have already occurred. You don't need to write any of this down. This is just an introduction to what we're going to talk about. But it's a pedigree, right? So geneticists for humans, we just look, instead of forcing people to mate, we just look at matings that have already occurred, and we can study that. And that is what's called a pedigree. Now, a pedigree can do a lot of things, but for some reason your book points this out, singles this out, but a pedigree can be used to determine if a trait is sex-linked, meaning it's on the sex chromosomes as opposed to any of the other 22 chromosomes. Now, I'm going to talk a little bit about that, but not much. To be honest, the sex-linked thing is a little bit more complicated than the other stuff. And also, in the grand scheme of this semester, it's not that important, so we're just going to skim over it. Anyway, the next picture I'm going to show you shows a pedigree tracing the incidence of free versus attached earlobes, which is what we've talked about, right? And Mendel's law will enable us to deduce the genotypes. You might get a question like this on the exam. So here we go. We have a bunch of phenotypes here. If it's purple, colored in, then that means you have attached earlobes. And to remind you, attached earlobes is recessive. So here we go. At least we know those genotypes, right? If they have the recessive phenotype, we know, just like the black or brown Labrador, we know their genotypes. They have to be homozygous recessive. Little e. There is no other possibility here, because if they had a big E, that looks so horrible, then they would have the dominant phenotype. So there we go. We know some of these genotypes. And using what we know about genetics, we can fill in some of the other blanks. That's so horrible. Okay. There we go. Now, let's think about what we know. And you might get something like this on the exam. Not because this is important that you be able to ever do this in your life, especially as non-biology majors, but the ability to think about this and understand it really shows that you understand genetics, which is important. All right. So here we go. Let's look at this one. we know that this person has a homozygous, or excuse me, has the dominant phenotype. So we know their genotype is big E something, right? We don't know what the other big E is. Could you guys agree with that? We know they have at least one big E. This person too. We know they have at least one big E. We don't know what the other allele is. Now, the fact that this person, their offspring, homozygous recessive, means that they got one allele, one little E from one parent and one little E from the other parent. You guys with me so far? Does that make sense? Like you have to, you get both alleles from your parents. Therefore, we know that this person has to be heterozygous. Because if they were homozygous recessive or dominant, if they only had, if it was big E, they couldn't have that child because they're only giving big E's and they have a child that has two small E's. So that person has to be heterozygous. Right? Otherwise, they couldn't have a homozygous recessive individual. Now, this one's a little bit trickier. If you were to just look at this, basically to figure this out, you would need to do a Punnett square, which I'm not going to do. I'm just going to remind you. I'm just going to start it, not finish it. Right? You would have a little e crossed with a big E, little e. And if you were to work that out, you would see there's a 50-50 chance, right, of one over the other. So that in itself does not give you the answer for this. But here we go again. We know because this person has a homozygous recessive child that they have to be heterozygous. There's no way for this person with this dominant phenotype to have a child with a homozygous recessive genotype, with a recessive phenotype. They have to have at least one small allele. Same with this person. There we go. We figured that person out too, right? There has to be, each one of them has to have a small allele to give in order to have a child that is homozygous recessive. So we also know this person is big E, little e. Now, you can go on and on. I'm not going to waste your time doing this because, again, you might have a question like this on the exam. If you do, it'll most likely be extra credit. And or maybe I'll post some stuff like this for independent work. But because it's a 50-question or a 50-minute exam, I'm not going to take a bunch of your time and ask you to do something like this. But anyway, that's how it works. And if we had the time, yes, we could figure out the other genotypes too. Any questions about that? And I know this is obvious, but to remind you, you don't need to memorize this. This is just an example. I'm not going to ask you. There's not going to be a question on the exam like, hey, remember that pedigree? Yeah. What color, what was the purple? What was that for? Freckles or attached earlobes, right? I'm not going to ask you that. Anyway, speaking of which, who cares about freckles and attached earlobes and cleft chins, right? That's not that important. So let's talk about something that is a little bit more important, using this information that's for something that's important. We'll talk about human disorders controlled by a single gene. So here we are. We're still talking about phenotypes. And instead of talking about, hitchhiker's thumb or straight thumb or bent little fingers or not, or hairy little knuckles, we're talking about something important, human disorders. And this is not hypothetical. This is important stuff. Some disorders are known to be dominant or recessive and controlled by a single gene. Spoiler alert, most of them aren't. So this is kind of rare, but it makes for good studying because when it's controlled by a single gene, it's very simple, right? You either have it or you don't. And the ones we're going to talk about are located on the autosomes, right? The ones other than the sex chromosomes, because we'll talk about sex-linked stuff later. That makes things a little bit more complicated. But here we go. Here's some examples. I'm not going to ask you any questions about this. You don't need to study this table. This is for your own knowledge. Just some examples of some, and we're going to talk about some of these. Some recessive disorders like albinism, cystic fibrosis, PKU, sickle cell disease, Tay-Sachs disease. Some dominant disorders such as dwarfism, Alzheimer's, that's a big one. Huntington's disease, hypercholesterolemia. You can look these things up for independent work too if you want to write any more about them. All right. Let's talk about recessive disorders first. Most disorders are recessive, at least when we're talking about disorders caused by a single gene on a non-sex chromosome. That's what the little star is about. And we'll talk more about them later, right? So when we come back, or when we talk about dominant disorders, we're going to talk about why most of them are recessive. But for now, just know that most of them are recessive. Not that I'm going to ask you that on the exam, but there you go. Individuals with a recessive allele, right? So if they have one recessive allele, but not two recessive alleles. They're called carriers. You do need to know that. A carrier, if we're talking about a recessive disorder, is somebody who has one of the recessive alleles but does not have the disease, which makes sense because, again, it's a recessive disorder. It's a recessive phenotype. So just like any other recessive phenotype, you have to have two recessive alleles to have that phenotype. Or another way of saying that, and this will be important a little bit later, instead of saying you have to have two recessive alleles, which is true, another way you could say it is 100% of your alleles have to be recessive for you to have that recessive phenotype. The same is true for these disorders. So if you're heterozygous, the other word for that is you're a carrier. If we're talking about disorders, that means you're a carrier. I mean, I guess you could almost apply that to any other thing too. Like my mom has brown eyes, my dad has brown eyes, but I have blue eyes. So technically, obviously, each one of them had to have that recessive allele. So you could call them carriers. They were carriers of blue eyes. Anyway, we can predict the fraction of affected offspring that are likely to result from the mating between two carriers. That's what we're going to do on this next slide. So here we go. Let's do this hypothetical hearing disorder. It's a recessive disorder, which means 100% of your alleles have to be recessive for you to have it. And here we have two people mating, and they're both carriers, right? They're both heterozygous. They both have normal hearing because they have that dominant allele that makes everything okay. But they also have that recessive allele that they can pass along. So we just do a regular old Punnett square that you should be very familiar with, especially when we're dealing with two heterozygotes. And here's the possibilities, right? And this is, again, the kind of test question you might get. Like I mentioned this earlier, I'm bringing it back. I might say this person mates with this person. What are the chances of their kid being able to hear or not being able to hear or being a carrier, right? So you would need to do this work right here. So there's a 25% chance of them hearing. There's a 50% chance of them hearing but being a carrier. And there's a 25% chance of them being deaf. You guys can see that, right? Hopefully that makes sense. And again, like I said earlier, when I talked about the rules of probability, to make it a little bit more challenging, I might say, what are the chances of Bob and Diane, and I'll give you all this pertinent information, like, oh, it's a recessive disorder, they're both carriers, whatever. What are the chances of Bob and Diane mating and having a boy who's a carrier, right? So that's a 1 in 2 chance, or excuse me, keep it simple. Yeah, what are the chances of having a boy who's not a carrier and can hear, right? So 1 in 4 chance of that happening, right? There's a 1 in 2 chance of having a boy over a girl. So you would multiply those. So the answer would be there's a 1 in 8 chance. Make sense? All right. So again, there will be a couple of questions where I give you this pertinent information, and you have to figure out what are the chances of this offspring. What are the chances of that offspring? Not many, so don't stress yourself about it. Some people are just really bad at math or really afraid of it for whatever reason. So if that's you, take a deep breath and don't worry about it. It's just one question or very few questions. Really quickly, let's use this example and talk about cystic fibrosis. I'm not going to ask you any questions about it, but your book talks about it, so I will too very quickly. It is the most common lethal genetic disease in the United States. So if you wanted to, that's a good independent work topic. First of all, I'm barely going to tell you about cystic fibrosis. You can look into it some more if you want. Also, what is the most common lethal disease, genetic disease in other countries? Or what about worldwide? Because your book specifically says in the U.S. It's caused by a recessive allele carried by about 1 in 31 Americans. And there you go, right? That's all I'm saying about cystic fibrosis. Really quickly, yeah, we can run through this quickly. Let's talk about dominant disorders. So dominant disorders are just like dominant phenotypes, right? You only have to have 1 allele to express it. So unlike recessive disorders or recessive phenotypes where all your alleles have to be recessive, with dominant, you only need 1. Your book is an interesting example, which is called achondroplasia. It's a type of dwarfism, which that actor right there, this was what he has. But this one's a little bit more interesting because, again, you only need 1 allele to have the disorder, right, because it's dominant. But this one's got a little twist to it. In this case, if you're homozygous dominant, you don't necessarily have the disorder because you're dead, right? If you're homozygous dominant, you're not even born, you die before birth, right? So the only people who have achondroplasia are heterozygous, because if they were homozygous dominant, again, they just wouldn't exist, they'd be dead. And if they were homozygous recessive, they wouldn't have it. So all the people who have achondroplasia are heterozygous. And because of that, and I'm not going to show you the math because it doesn't matter, but you could figure it out on your own and do some Punnett squares. You can see that a person with achondroplasia has a 50% chance of passing that condition on to their children, because we know it's dominant, first of all, and then we know that they're I am going to show you the math, and we know that they're heterozygous. Because again, in this case, they have to be. If they were homozygous dominant, they would be dead. If they were homozygous recessive, they would be normal. So anyway, that's what we mean by them having a 50% chance. Now, if you want to, for independent work, answer this easy question. What are the chances of a dwarf and another dwarf having a dwarf child? You just do the same thing. Do a Punnett square, except instead of heterozygous and homozygous recessive, you can do heterozygous versus heterozygous. And tell me, what are the chances of them having a dwarf as a child? And what are the chances of their child not being born at all? In other words, what are the chances of them having a homozygous dominant child? Anyway, dominant lethal disorders are much less common than lethal recessive disorders. This should make sense, because if it's lethal and you get it, then that means you're going to die, right? And it's really hard to breed, pass along your genes after you're dead, right? Your book gives this example. Huntington's disease is a great example. That's a degeneration of the nervous system. What makes it relevant to this conversation is that the symptoms don't usually start until middle age, meaning you could carry this deadly disease and still be alive to pass along the genes, right? So even though it's deadly, you could still pass along the genes. And that's basically all you would need to know about this on the exam. And I probably won't ask you that anyway. But if anything, yeah, I'm not going to ask you anything about Huntington's disease. But again, the fact that you don't see symptoms until later in life is how it's passed along. And because the allele for Huntington's disease is dominant, any child born to the parent with the allele has a 50% chance of inheriting it themselves. And that's if only one parent has Huntington's. Imagine if they both had Huntington's. Anyway, again, that's just an example. I'm not going to ask you anything about that on the exam. The last word for attendance will be dogs. So again, I'll remind you, if you're online, whether you're watching the video or the live stream now or watching the video later, send me pictures of all those words instead of the actual words. And the rest of you can just send pictures or the words if you want for extra credit. So any questions? All right, you guys have a great weekend.
Okay.